Assalamu alaikum. Good morning, everybody. Today we are going to start our first physiology lecture in the elementary module, and the title of today's lecture is the Physiology of Esophageal and Gastric Motility. As you might remember from the introductory course, the digestive system is composed of several organs that can be categorized under two main groups. These are the gastrointestinal tract or the alimentary canal. The abbreviation is GIT. It is the tube that extends between the oral cavity and the anus, the end of the large intestine. And these organs, as you can see here in this figure, are the oral cavity, the pharynx, esophagus, stomach, the different parts of the small intestine, and the large intestine. And the second group of organs are the accessory digestive organs, and these are the teeth, tongue, salivary glands, liver, gallbladder, and the pancreas, and they are of great help in the process of digestion. The organs of the digestive system can perform one or more of the 6 basic digestive processes that you have learned in the introduction course. These 6 basic processes are ingestion, which is the intake of food and liquid into the oral cavity, and then secretion, and these are watery secretions produced by glands present in the mucosa of the alimentary canal or its submucosa, as well as the accessory digestive organs, and they are of great help in performing or helping the digestive process. Then the third basic process is the propulsion and mixing, and propulsion means pushing food and liquid from the oral cavity to a more distal direction towards the anus. The fourth important basic process is digestion, and it is composed of two parts: mechanical digestion, which is the function of the muscle fibers in the muscularis externa, which is part of the wall of the alimentary canal, and we have another type of digestion, which is the chemical digestion, and it is a function of enzymes present in the digestive secretions or in enzymes embedded in the apical membrane of the epithelial cells lining the small intestine, and we call them the brush border enzymes. The fifth and the most important process of digestion is absorption, and this is the ultimate goal of the digestive process, where our body can absorb the nutrients, minerals, vitamins, water that we intake them during a meal. And finally, what is left over that cannot be digested will be eliminated from the body by the process of defecation. Let's start with the esophageal motility. As I mentioned earlier, any organ in the digestive system can perform one or more of the basic digestive processes. And let's talk about the esophageal motility, because the esophagus mainly serves as an organ for propulsion or the transport of food bolus from the oral cavity through the pharynx down the esophagus to the stomach. The objectives are to describe the functional anatomy of the esophagus and its innervation, understand the roles played by the esophagus in transferring food from the pharynx to the stomach during swallowing, define the mechanisms and functions of primary and secondary esophageal peristalsis, which are the main motility patterns in the esophagus, describe the roles played by the upper and lower esophageal sphincters in esophageal motility, and discuss disease states in which esophageal motility and or the swallowing process are abnormal. The esophagus is a muscular tube that serves to transfer food from the mouth to the stomach. It has a length of about 18 to 25 centimeter, and the length varies with the height of the person, so tall people have longer esophagi and the opposite is true. Like other parts of the alimentary canal, the wall contains the third layer, which is the muscularis externa, and the muscularis externa is composed of two muscle layers that have different arrangements. The innermost layer is circularly arranged, and we call it the circular layer, and the outermost layer is longitudinally arranged, and we call the longitudinal muscle layer. Unlike all more distal segments of the gastrointestinal tract, the esophagus has a unique composition of or unique type of muscles in its muscularis externa. The uppermost part is composed of striated muscles. The lower third is composed exclusively of smooth muscles, and the middle third is a mixture of striated muscles and smooth muscles, and these different parts or thirds of the esophagus have different patterns of innervation, especially in terms of motor innervation. At rest, the esophagus is a relaxed structure that is closed off at both ends by two sphincters, and these are the upper and lower esophageal sphincters respectively. These sphincters, or these two sphincters, not only cooperate in the act of swallowing or deglutition, but also prevent the backflow of gastric contents into the esophageal lumen or oral cavity. And this backflow could happen because the esophagus is situated within the low pressure thorax, or the thorax that has a negative pressure, and thus the presence of these two sphincters is important to prevent the entry of air and gastric contents into the esophagus at rest. Under normal conditions, food will remain in the esophagus for only a few seconds, and thus there is no time for it to be acted upon by any esophageal secretions, so the esophagus doesn't store food and doesn't digest food because the transit of food bolus through it is very fast. Understanding the physiology of the esophagus relates primarily to its motility function. As I said earlier, the main digestive process that happens in the esophagus is propulsion, and this is the function of motility. In addition to moving the food along its length in the process of swallowing, the movements of the esophagus and related oral and pharyngeal structures must be carefully coordinated or regulated to avoid misdirection of food into the respiratory tract or the misdirection of respired air into the stomach or the remaining parts of the digestive system. Under specific conditions, the esophagus does not allow for retrograde movement. Retrograde movement means movement of food or any contents from the stomach back to the pharynx and oral cavity. This occurs normally for air swallowed with meal in the process of belching, or abnormally during vomiting. So retrograde movement of air is normal and it is called belching, and retrograde movement of gastric content through the esophagus to the pharynx and oral cavity is called vomiting. During the retrograde movement in humans and most mammals, the esophagus itself is a passive conduit. That is, there are no specific motility functions that propel the vomitus or air along the length of the tube, which means that the esophagus during vomiting is completely relaxed. There are no contractions or no peristalsis or retroperistalsis that happen in the esophagus during vomiting, and I will stress on this concept at the end of this lecture when we will discuss the process of vomiting. Esophageal motility is mainly in the form of peristalsis as a part of the sequentially programmed all or none swallowing or deglutition reflex that has three different stages. One of them is voluntary and the other two are involuntary. The first stage is the oral or buccal stage that happens in the oral cavity to push the food towards the pharynx. The second stage is the pharyngeal stage, where food will pass through the pharynx down to the esophagus, and it is involuntary. Then the final stage of swallowing reflex is the esophageal stage. Again, it is involuntary, and in that stage the esophageal peristalsis pushes the food bolus towards the lower esophageal sphincter, that in turn will relax to allow the bolus to enter the stomach. As you can see here in these two figures, we have the oral stage, the pharyngeal stage, and finally the esophageal stage. The end of the esophageal stage, the lower esophageal sphincter will relax and will allow the food bolus to enter into the stomach. In health, these stages of deglutition are tightly integrated and very well coordinated, so the swallowing reflex goes or happens in a very smooth way. Now let's talk about the types of esophageal peristalsis. The first type is the primary peristalsis. Once the bolus has moved through the upper esophageal sphincter into the esophageal lumen, it is moved along the length of the tube by primary peristalsis. The primary peristalsis starts at the upper esophageal sphincter and ends by the relaxation of the lower esophageal sphincter to allow the food bolus to enter the stomach, and it is a series of coordinated muscular contractions and relaxations. The sequencing and thus the direction of these or of this propulsive process is coordinated, with contraction of more distal segments occurring at longer latencies or occurring later on following the swallow than in those close to the pharynx. As you can see here in this figure, we placed pressure sensor and we are recording something called the esophageal manometry. We record the pressure along the esophagus. As you can see here, the pharyngeal stage, there is contraction, and we can notice that there is an increase in the pressure in the pharynx, and this is followed by, or at the same time, with relaxation of the upper esophageal sphincter. This negative deflection here in the figure shows relaxation, and then followed by the contraction of the upper esophageal sphincter to prevent the return of the food bolus back to the pharynx. And then the different segments of the esophagus will sequentially contract until the bolus reaches the lower esophagus, where the lower esophageal sphincter will relax, as we can see here in the figure, to allow the food bolus to enter into the stomach. The primary peristalsis, this means that the ability of the body to transfer food from the mouth to the stomach is largely independent of body orientation. So we don't depend on gravity to swallow food, or in our primary peristalsis we don't depend on gravity. However, gravity will help liquids. Gravity can be an important determinant of transit rate, particularly where liquids are ingested. They have little effect on swallowed solids. Liquids may reach the lower esophageal sphincter before the peristaltic wave has reached that site or the lower end of the esophagus. The peristaltic wave requires about 10 seconds to pass from the upper esophageal sphincter down to the lower esophageal sphincter. And this is the time needed by solid esophageal content to sweep down from the pharynx to the stomach. The other type of peristalsis is secondary peristalsis. In secondary peristalsis, it is a wave that does not start in the upper part of the esophagus, and this is the difference. The secondary peristaltic wave is a function of the smooth muscles present in the wall of the esophagus, and as we know, the upper third of the esophagus is composed entirely of striated muscles. So the secondary peristalsis doesn't happen in the upper third. It could start in the middle third, as the middle third contains smooth muscles in addition to striated muscles, as I mentioned in the earlier slides. The primary wave of peristalsis along the length of the esophagus may also be followed by secondary wave that is restricted to the smooth muscle portion. It means it can start from the middle third, and also it can occur in the lower third. Secondary peristalsis is likely to be important in clearing a bolus that was not wholly expelled from the esophagus during the primary peristaltic wave, or in removing any gastric contents that reflux back into the lower esophagus from the stomach into the lower part of the esophagus, and this is called the gastroesophageal reflux, as we'll study in one of the tutorials. So as we see here in the esophageal manometry, you can see that there are no contractions, and the pressure sensors or the manometer will not record any contractile waves, and there are no pressure changes in the striated esophagus. The contractions start in the mid-esophagus down the lower esophagus to clear any leftover from the previous primary peristalsis, because sometimes the primary peristaltic wave cannot completely clear or push the food bolus into the stomach, so the secondary peristaltic wave will do that function. The peristaltic response, or the secondary peristalsis, is triggered by distension that is caused by the remaining food bolus, and involves both local reflexes, so the entire process or reflex happens in the wall of the esophagus, and we call it local reflex within the enteric nervous system, as well as the long central reflexes, and we are talking about the vagovagal reflex, which is the most important long reflex in the digestive system. The presence of acids can also trigger secondary peristaltic wave. So any regurg of acid from the stomach into the lower esophagus can trigger secondary peristaltic wave. So the presence of acid alone within the distal esophagus in the absence of significant distension can also, or is also, sufficient in generating a secondary peristaltic response. And here the reflex is mediated by chemosensitive afferents rather than by mechanosensitive afferents, as in the case of distension. So what stimulates the esophageal peristalsis? Peristalsis in the esophagus is stimulated either by distension or by chemicals, and when we talk about chemicals, we mean mainly the pH, the acid from the stomach. And these stimuli, or these mechanical or chemical stimuli, will stimulate, as you see here, a visceral sensory fiber that will carry the information to the dorsal vagal complex in the central nervous system as part of the long reflex. From the dorsal vagal complex, efferents or visceral motors or the parasympathetic fibers will run down the vagus nerve to the alimentary canal or to the esophagus in this example, and this will lead, or these vagal efferents will synapse with enteric neurons. The enteric neurons proximal to the bolus are cholinergic, and the acetylcholine will cause contraction proximal to the bolus, while the enteric neurons or the myenteric neurons that are distal to the bolus are nitrinergic, and they release nitric oxide that will cause the relaxation of the esophageal wall, and this will help the propulsion of the food bolus down the esophagus to the stomach, and this is the long vagovagal reflex. The other example here in the figure on the right is how local reflex are triggered to cause propulsion or secondary peristaltic wave. Here distension or low pH in the lumen of the esophagus will be processed locally at the level of the myenteric plexus. As we can see here, we have interneurons that transmit the signal between the afferents and the efferents. So the excitatory enteric motor neurons will innervate or will stimulate muscle fibers proximal to the food bolus via the release of acetylcholine to cause contraction of the circular muscles, while the enteric neurons or myenteric neurons distal, supplying the part distal to the food bolus, will release nitric oxide or VIP, vasoactive intestinal peptide, and both of them, they cause relaxation of the circular muscles, and this will ease the propulsion of food bolus down a relaxed portion of the esophagus. So mechanoreceptors or chemoreceptors on sensory afferents transmit impulses to the dorsal vagal complex, which in turn activates vagal efferents that terminate either directly on the striated muscles, and this happens in the upper part of the esophagus where it is composed mainly of striated muscles, or onto nerves of the enteric nervous system in the lower third. These enteric neurons activate vagal fibers innervating the enteric neurons will stimulate enteric neurons that release acetylcholine proximal to the food bolus or nitric oxide distal to the food bolus. The esophagus also transmits information about its content back to more proximal segments. The presence of water, or more potently acid, in the esophagus increases the pressure of the upper esophageal sphincter to prevent their backflow into the pharynx and oral cavity. The intensity of this stimulation increases as one moves closer to the upper part of the esophagus, and the reflex presumably presents a protective mechanism to restrict esophageal or refluxed gastric contents from returning to the pharynx, from where they might otherwise be aspirated and cause trouble. On the other hand, if air is present in the esophagus, it induces the opening of the upper esophageal sphincter, which is critical to belching, or the retrograde movement of air that is swallowed along with food. And still today, nobody knows how or what mechanism discriminates between air and liquid, because we said liquid will cause closure of the upper esophageal sphincter, while air causes the opening of the upper esophageal sphincter, and we don't know the exact mechanism, why they work in different ways. Another motility pattern in the esophagus is the lower esophageal sphincter relaxation, or LES relaxation. The final component of esophageal motility is relaxation of the lower esophageal sphincter, allowing the food bolus to move into the stomach. And the resting condition, the lower esophageal sphincter is tonically contracted because it has a myogenic tone independent of a neural input, and it increases intrinsically as it is stretched. The basal tone of the sphincter protects the lower portion of the esophagus from the corrosive effect of the gastric juice, especially those mediated by the acid, the gastric acid or the HCl, and pepsin, the protein digesting enzyme pepsin. The squamous epithelium lining the esophagus is very sensitive to these components, so it is much less able than the gastric epithelium to defend itself from these injurious substances. The tone of the sphincter can be increased by neurohumoral agents released in concert with the ingestion of a meal, including acetylcholine and gastrin. The extent of this effect is dependent on the composition of the meal, and with proteins increasing the sphincter tone to a greater extent than fat. So the presence of protein in the stomach can cause the release of gastrin. Gastrin will cause the constriction or increase the tone of the lower esophageal sphincter. LES pressure is substantially augmented or helped by the crural diaphragm, which wraps around the lower part of the esophagus. This also accounts for the increase in LES pressure during inspiration and also during coughing. In healthy individuals or subjects, the lower esophageal sphincter relaxes transiently from time to time in a manner that is independent of either swallowing or secondary peristalsis. These responses are triggered by gastric distension, which in turn activates vagovagal pathways that reduce the tone of both the LES and the crural diaphragm via nitric oxide and VIP. Such relaxations facilitate belching of swallowed air and are more common when we are upright, which presumably protects against acid reflux to some extent. In individuals with reflux disease, the transient lower esophageal sphincter relaxations are more frequent, and this is associated with reflux of gastric contents into the distal esophagus, as we'll see in the GERD tutorial. Here we discuss two pathological conditions that affect the motility pattern of the esophagus. One of them is achalasia. It is a special form of dysphagia and means failure to relax. This condition occurs when the lower esophageal sphincter does not open fully as part of the peristaltic wave that sweeps bullous down the esophagus into the stomach. And the failure of the LES to relax is usually due to degeneration of cell bodies in the myenteric neuron that release nitric oxide and VIP. And this could happen as part of an autoimmune attack to those cells, or as part of the parasitic infection like in Chagas disease, where the parasite attacks cell bodies in the ganglia, including those of the esophagus, as well as other systems in the body like the heart, urinary tract, respiratory tract, and other segments of the alimentary canal. Another example of a pathophysiology that affects the motility of the esophagus is the gastroesophageal reflux disease or GERD that we will discuss thoroughly in the tutorial, inshallah. Now let's go to the gastric motility. The objectives of this part of the lecture are to understand how the motility functions of the stomach contribute to the integrated response to a meal, describe the muscle layers of the stomach, their connections to the enteric nervous system that observe gastric motility, define the motility patterns that characterize movements of the stomach under fed and fasted conditions. We mean fasted conditions, the interdigestive period or between the period between meals, and their control mechanism. Describe receptive relaxation and mixing or grinding patterns of the motility and the regulation. Understand how the stomach is emptied and how this is coordinated with the function of downstream segments. Understand the pathophysiology of vomiting. The stomach is a muscular reservoir into which the meal enters after being swallowed. It is a particularly distendable organ. The volume of an empty stomach is 50 ml, and it can increase up to 1 liter, which is 20 times larger compared to the empty stomach. It can be considered as homogenizer, mechanically breaking down ingested food into an emulsion of small particles with a vastly increased total surface area, thereby amplifying the effect of digestive enzyme and help the process of digestion. It is a critical contributor to the matching of food delivery to the digestive and absorptive capacity of more distal segments of the gut. The duodenum cannot deal with the entire meal after we ingested, so the meal will be stored in the stomach, and then the stomach will continually mechanically and chemically digest that meal and release a small amount of chyme to the duodenum, amounts that are small enough so the duodenum can deal with them without any problem. The stomach thus serves as a reservoir, allowing food particles to move only slowly into the duodenum to maximize their chances for assimilation, and we mean the completion of digestion and then absorption. Distension of the stomach also delivers important information to the downstream segments of the gastrointestinal tract, and a good example is the gastrocolic reflex distension of the stomach causes contraction in the colon and can elicit defecation reflex, and also distension of the stomach can contribute or contributes signals to the satiety center. The stomach has the same two main layers of the other parts of the alimentary canal. These are the innermost circular layer and the outermost longitudinal layer. In addition to that, a small region of the stomach contains obliquely oriented muscle fibers, which is the innermost layer here in this case, and is present in the lesser curvature of the stomach that is continuous with the gastroesophageal junction. As you might remember from the anatomy lecture, the stomach is divided anatomically into four parts: the cardia, the fundus, the corpus or the body, and the pyloric region. The pyloric region has an antrum, a narrow pyloric canal, and the pylorus or the pyloric sphincter. The pylorus is a specialized region of circular muscle fibers at the point where the caliber of the gastric lumen is sharply reduced prior to entry into the duodenum. It serves as a mechanical barrier to food exit that is also enhanced by folded redundant mucosa, as we can see here, the folds of the mucosa. The stomach can be divided from the physiological point of view into two main regions based on the difference in the motility pattern in each region. As we can see here in this figure, we have a proximal part and a distal part. The proximal part is characterized by tonic contractions and it has reservoir function, and the distal part is characterized by phasic contractions, and it is called also the antral pump. It is very important for the mixing, grinding, and the gastric emptying. So the proximal stomach consists of the cardia, fundus, and proximal portion of the body of the stomach. It serves primarily as a reservoir and also moves gastric contents to the distal stomach. Tonic contractions of this part are important in gastric emptying. The distal stomach consists of the distal portion of the body plus the pyloric antrum. It serves predominantly to grind and triturate the meal by phasic contractions. The pylorus acts as a sphincter that controls the amount and size of food particles that can exit the stomach in the fed state or during meal. As I said, the stomach is divided from the functional point of view into two parts because the gastric smooth muscles exhibit two distinct myogenic contractile properties that are intrinsic to these cells and independent of either neural or humoral input. There are cells that exhibit tonic contractions, which are sustained contractions that may persist for many minutes, and these type of muscles are prominent in the proximal stomach. We have smooth muscles that exhibit phasic contractions, where the smooth muscles contract and then relax in a matter of seconds, and these types of muscles are predominant or more prominent in the distal stomach. Each type of contraction is important in mediating the specific motility property that are needed for the function of each region of the stomach, either proximal or distal, as we explained earlier. The gastric musculature is under both extrinsic and intrinsic innervation. Extrinsic innervation is mainly via the parasympathetic fibers present in the vagus nerve and is the major extrinsic pathway for the regulation of gastric motility. Most vagal efferents are cholinergic. They are stimulatory because they release acetylcholine, causing the contraction of smooth muscles. Few parasympathetic fibers or vagal efferents are inhibitory because they release NO and VIP. Vagal afferents or vagal sensory fibers are also critical for the control of motility functions. These are of both mechano and chemosensitive type, so they can be stimulated by mechanical stimuli and chemical stimuli. Stretch and chemicals mainly hydrochloric acid or hydrogen, and activate sites in the nucleus tractus solitarius of the dorsal motor nucleus in the brain, and they are part of long central reflexes. Sympathetic innervation via the splanchnic nerve are less important in controlling gastric motility, and they are inhibitory at the level of the enteric ganglia because they release norepinephrine. The physiological role of sympathetic innervation to the stomach in producing the motility pattern that characterize the response to a meal is minor compared to the vagal influence. So the main extrinsic influence on the stomach is parasympathetic via the vagus nerve. In terms of intrinsic innervation, it is via the enteric nervous system, mainly the myenteric division, which is critically important to the full expression of gastric motility responses. Myenteric neurons of the stomach also coordinate gastric motility functions with those of more distal segments of the gut, and an example is the gastrocolic reflex that I mentioned earlier. These nerves also communicate with the pacemaker cells of the gut. We call them the interstitial cells of Cajal. They are specialized smooth muscle cells that have pacemaker potential. Spontaneous depolarization, they are similar to the specialized cardiomyocytes present in the heart, and that represent 1% of the total number of cardiomyocytes, and we call them the autorhythmic cells. This communication between the myenteric neurons, interstitial cells of Cajal, and the smooth muscles of the stomach or the contractile smooth muscles establishes the rate at which contractions of the tissue can maximally occur if an additional excitatory signal is also received. And this is determined by the existing basal electrical rhythm or the slow wave potentials that I will explain in the coming slides. Here we can see a figure showing the different layers of any part or segment of the gut wall. We have the four basic layers: the mucosa, submucosa, muscularis externa, and serosa or adventitia in certain parts. The muscularis externa or the muscular coat is divided into innermost circular layer, outer longitudinal layer separated by the myenteric plexus, which is part of the enteric nervous system. We can see here a fine network of the specialized smooth muscle cells called interstitial cells of Cajal, and here they are called the myenteric interstitial cells of Cajal because they are associated with the myenteric plexus. The basal electrical or electric rhythm, BERs, are waves of depolarization that originate in the pacemaker cells of the gut or the interstitial cells of Cajal at a specific point, and they spread to adjacent smooth muscle cells via gap junctions. These pacemaker activities are not of sufficient magnitude to initiate action potentials in the smooth muscle. So we have to add to potentiate the signal to reach the threshold and have full-blown all or non-action potentials. So when stimulatory neurotransmitters or hormones are released from the enteric nerves, are superimposed on these waves of depolarization, the threshold for contraction of the smooth muscles will be reached, and we can see here that these BER at rest do not generate any tension or any contraction. When additional signals are added, for example acetylcholine released from the myenteric neurons, threshold is reached, action potential will happen, and this will lead to contraction of the smooth muscles. Here we can see again the membrane potential, and the resting state we have these pacemaker potentials, and then when the stimulation by stretch, acetylcholine or the parasympathetic system, this will lead to spikes. With increasing stimulation, the spikes become more frequent, and this will lead to depolarization and contraction. Inhibitors like norepinephrine or stimulation of the sympathetic nervous system will cause hyperpolarization, and the pacemaker potential will disappear. In the stomach, the pacemaker potentials appear to begin at a point in the body along the greater curvature of the stomach, just between the or at the junction of the proximal part and the distal part of the stomach, and then they sweep down across the stomach towards the pylorus. The pacemaker potentials originate at this point, determine the contractile pacemaker of the stomach as a whole, namely the maximal frequency of contractions, their propagation velocity, and the direction in which they propagate. The BER differs in various intestinal segments or in different segments of the alimentary canal. For example, in the stomach, the BER happen at approximately a rate of 3 cycles per minute, in the duodenum 12 cycles per minute, and 9 cycles per minute in the ileum, and 3 cycles per minute in the colon. And these decrement or decrease in the number of cycles as we go down from the duodenum to the colon is important in the propulsion of chyme to more distal regions, as we'll see in the small intestine lectures. The stomach exhibits different motility patterns. We have motility patterns during meal or during the fed state, and motility patterns during the fasting state or the interdigestive state. The motility patterns during the fed state are receptive relaxation, mixing and grinding, and gastric emptying. The motility pattern, or the main and only motility pattern that happens during the fasting or interdigestive state, is the migrating, or also called migratory motility complex, the MMCs. The receptive relaxation, the ability of the stomach to relax as its volume increases, is essential to its reservoir function. It results in a drop of gastric pressure immediately after eating that persists until all solids have been emptied from the stomach. It is divided into two phases. One of them is the true receptive relaxation. The relaxation will happen before the arrival of food bolus into the stomach, and it happens or occurs coincident with the swallowing as part of the swallowing reflex. The other type or phase of receptive relaxation is accommodation. It is the relaxation of the stomach that is mediated by the presence of food bolus in the stomach that will stimulate mechanoreceptors in the wall of the stomach by stretch, and this will lead through local, short, and long central reflexes to the relaxation of the muscles in the wall of the stomach. And again, as you can see, the vagus nerve can stimulate enteric neurons that release nitric oxide and VIP, and also local reflexes can also stimulate enteric motor neurons that finally lead to the release of nitric oxide and VIP, and both of them can relax the smooth muscles of the stomach. The other motility pattern is mixing and grinding. It happens, or it is the primary motility pattern of the distal portion of the stomach during the fed state, and consists of rapid phasic contractions that occur circumferentially. The rate of these contractions depend on the gastric basal electrical rhythm, and they occur when the release of contractile agonists is superimposed on this rhythm, thereby increasing the magnitude and duration of individual contractions. The contractions proceed from the gastric pacemaker region and moves toward the pylorus in a peristaltic pattern. As these waves of contraction begin, they force the gastric content towards the outlet of the stomach, and there only small sized particles will pass through the partially open pyloric sphincter. As we can see here in this illustration, we have phasic contraction A, that started at the pacemaker region of the stomach at the junction of proximal and distal parts of the stomach and swept down the pylorus. And after a while, another phasic contraction will start at the same site and sweep down, and then another one, and so on. With each contraction, small amount of chyme will be emptied into the duodenum, and the rest will be returned back to the body of the stomach by retropulsion. The majority of the meal is forced back towards the body of the stomach. As I said, the net result of this backward movement or retropulsion is to mix the gastric contents thoroughly with the gastric juices and to mechanically reduce the size of food particles. Mechanical dispersion of the meal is also promoted by impelling the smaller particles against the largely closed pylorus, causing them to be acted on by shear forces or mechanical forces. In effect, the stomach acts as a sieve as well as homogenizer, gradually moving small particles of food closest to the stomach outlet or the pyloric sphincter, while retaining those that need to be reduced further. The third motility pattern of the stomach during the fed state is gastric emptying. And here the pylorus has very important role. When the stomach is filled with the meal, the pylorus remains closed for prolonged periods with only intermittent openings. The pylorus never relaxes completely during meal. This will allow small food particles to enter the duodenum. The pylorus is regulated by both inhibitory and excitatory vagal pathways, as well as ascending and descending intrinsic reflexes through the myenteric plexus. Nitric oxide has been identified as a key mediator of pyloric relaxation, arising from both vagal and intrinsic pathways, whereas opioids released from the vagus or the vagal efferents have been implicated in contractions. Also, hormones like CCK, that is released from the duodenum, may also increase the contractile functions of the pylorus in the postprandial period or the period after meal. Feedback reflexes from the duodenum also contribute to the pyloric closure, specifically the presence of nutrients, hypertonicity or acid in the duodenum. This will cause the pylorus to close. In addition to that, acetylcholine, serotonin are also mediators responsible for the closure of the pyloric sphincter. Gastric emptying is controlled by factors in the stomach, factors in the duodenum, and factors outside the gut. Factors in the stomach are the volume of chyme. If we have larger volume of chyme, this will stimulate motility and emptying. Also, the degree of fluidity of chyme. Increased fluidity allows more rapid emptying. Factors in the duodenum, the presence of fat will stimulate eye cells to release cholecystokinin from the duodenum, and this will circulate in the blood and inhibits gastric emptying. Acid in the duodenum stimulates S cells to release secretin that will inhibit gastric emptying. Hypertonicity and distension, both of them through mechano and chemoreceptors respectively, will stimulate the enterogastric reflex, and this reflex will inhibit emptying, while factors from outside the gut like emotions can inhibit or stimulate motility and emptying, while pain or intense pain will inhibit gastric motility and emptying. Out of these three main factors that control gastric emptying, the duodenal or factors in the duodenum are the main controller of gastric emptying. Now the main motility pattern that happens during fasting or interdigestive period. I said there is one main motility pattern, which is the migrating or migratory motility complex, or the MMC. In the period between meals, the stomach, in common with more distal segments of the GIT, and we mean here the small intestine, undergoes the special pattern of motility known as the migrating motility or motor complex, MMC. Cycles of motility lasting about 100 minutes and consist of three phases. Begin in the stomach and propagate orally or distally down to the ileocecal valve at the junction of small and large intestine. It is composed of three phases, as I said. Phase 1 represents about 40 to 60% of the cycle duration, and it is characterized by quiescence. There is no motility at all. Phase 2, contractile activity increases, but the contractions are irregular and fail to propel luminal contents, and it lasts about 20 or takes about 20 to 30% of the overall cycle period. The last phase, which is the most important, it lasts for about 5 to 10 minutes of intense luminal occlusive contractions that sweep from the body of the stomach to the pylorus and from there move down the duodenum. During this time, the pylorus opens fully in normal subjects, allowing any indigestible residues that remain in the stomach to be cleared from the lumen. So the stomach will be cleared from any undigested material in preparation for the next meal. Now let's talk about vomiting in brief. Vomiting is not a disorder of gastric motility per se. Vomiting reflects the coordinated interaction of neural, humoral, and muscular phenomena. Vomiting may be prompted by both central and peripheral triggers. So we have vomiting due to central triggers and vomiting due to peripheral triggers, but requires the involvement of central brain regions to coordinate the responses. A chemoreceptor trigger zone present in the area postrema of the medulla oblongata receives input from cortical, oral, vestibular, and peripheral afferents. In addition to the blood-brain barrier in this region is relatively leaky, so it can sample chemicals in the blood. This means that chemoreceptor trigger zone can sample chemical constituents of both blood and the CSF or cerebrospinal fluid. As we can see here, there are many triggers for the vomiting. These include irritation of the gastric mucosa by ipecac, cytotoxic drugs and irritants. This will stimulate the gastric mucosa. Information will be carried through the visceral afferents in the vagus nerve to the area postrema, or the chemoreceptor trigger zone, and the nucleus tractus solitarius of the vagus nerve. Also, triggers for the chemoreceptor trigger zone or direct triggers are drugs in the blood like opiate, chemotherapy, hormones like the hormones of pregnancy. Other stimuli are motion and vertigo that will be sensed by the labyrinth of the inner ear. Information will be carried to the cerebellum, from there to the brainstem. Pain, sights, and anticipation through higher centers. Information again will be carried to the brainstem, where the vomiting center is present. Stimulation of the pharynx. Information through the glossopharyngeal nerve will be carried to the nucleus tractus solitarius and then to the vomiting center in the brainstem. And then efferents from the brainstem and the nucleus tractus solitarius will lead to the programmed vomiting response. Various stimuli lead to both nausea, which is the sensation that vomiting is imminent or will happen soon, and actual vomiting. Thus, nausea and vomiting that occur secondary to endocrine causes, such as experienced in early pregnancy, are likely to be central in origin, as is vomiting provoked by noxious odors or smells, visual stimuli, somatic pain, or unpleasant taste. These are all central triggers of vomiting. Peripheral afferents can also trigger vomiting or trigger a vomiting response. This is especially the case when irritants are present in the gastric lumen and some vagal afferents originating in the stomach and presumably linked to the area postrema. Here we can see a table with different triggers of vomiting, like cancer chemotherapy, analgesics, antibiotics, narcotics, disorders in the GIT, like excessive stretch that is caused by mechanical obstruction, gastroparesis that we'll study in one of the tutorials, radiation injury to the gut wall, especially the mucosa, functional bowel disorders, intraperitoneal inflammation, and other causes of vomiting. and these include central as well as peripheral triggers of vomiting. Another brain region, the nucleus tractus solitarius, also contributes to the emetic cascade or the vomiting cascade, particularly following vagal activation, because vagal afferents will relay signals to the nucleus tractus solitarius, which is the sensory center or nucleus of the vagus nerve. This region receives input from the area postrema, abdominal vagus, and labyrinth, and in turn coordinates the motor responses required for vomiting. Emesis or vomiting results from sequentially programmed muscle actions. First, the thoracic, diaphragmatic, and abdominal muscles contract concurrently or simultaneously against the closed glottis, resulting in the phenomenon of retching. The lower esophageal sphincter and the crural diaphragm relax, and the high positive intra-abdominal pressure compresses the relaxed stomach, and this will force the gastric content into the esophagus. So during vomiting, the stomach is relaxed and the pressure is coming on the surface or the wall of the stomach from outside due to the contraction of the abdominal muscles and the diaphragmatic muscles. Then the brain coordinates the synchronous contraction of the inspiratory and expiratory muscles, resulting in a reversal of the thoracic pressure gradient. This high positive thoracic pressure acts in turn to drive expulsion of the vomitus. Simultaneously, respiration is suppressed, and movement of the larynx and pharyngeal structures prevents the aspiration of the vomitus, so it will prevent choking. Thank you very much for listening to this lecture, and here are the references from which I got the different informations related to the lecture. Thank you.